Good morning and welcome again to Grand Rounds. It is truly my pleasure today to introduce our Grand Rounds speaker, Dr. Andre Nell, who will be speaking to us about the development of a COVID-19 vaccination and nanomedicine. Dr. Nell is an internationally known expert in the field of nanomedicine. Dr. Nell completed his MD PhD training in South Africa in Cape Town, and then he came to the United States to complete his clinical training in allergy and immunology in South Carolina. In 1988, he joined the faculty here at UCLA, and for over 30 years, he has truly become one of the world's leading experts in nanomedicine. He's a distinguished professor of medicine, and he's the founder and chief of the Division of Nanomedicine at UCLA. He's the research director of the California NanoSystems Institute, and he's the director of the University of California Center for the environmental implications of nanotechnology. Today, Dr. Nell is going to speak to us on COVID-19 vaccination and nanomedicine. So it is my pleasure to turn it over to Dr. Andre Nell.
Okay, thank you, Jody, for the kind introduction. I hope everybody is keeping well amidst the topic that we would discuss today, namely COVID vaccination drawing on progress in nanomedicine. So nanomedicine is a discipline where we take an engineered approach to medicine with impact on therapeutics, diagnostics, imaging, and prevention. Whereas the field developed rapidly for treatment of cancer, infectious disease, and neurology, it is achieving more and more applications in other areas now, cardiovascular medicine, and regenerative medicine, as shown here on the left-hand side of the screen. What I hope today is to talk to you in terms of nanomedicine's impact on immunology, and particularly vaccines, which also draws from our work on tumor vaccination. And I hope that, as I'm frequently asked, what is nanomedicine, to be able to explain to you today the use of nanoparticles as a mimic of viral interactions with the immune system. So that's explained here. Nanoparticles in the immune system. Nanoparticles can be engineered to either avoid interaction or specifically interact with the immune system. This can be done on the left-hand side in a desirable fashion to obtain immune stimulation, and it could also have undesirable outcomes, or to deliberately promote immune suppression. That part I will not be talking about today. What nanoparticles can do in interfacing with the immune system is through the selection of the shape, the size, the charge of nanoparticles, and their ability to deliver things, is to develop communication with the immune system based on either delivery or danger signals. And by danger signals, I mean, as an example from our previous study, alum is a frequently used adjuvant in the immune system. It's a more like an amorphous powder. But if you make it into aluminum oxyhydroxide nanorods, those nanorods impart a much stronger stimulation by providing an immune danger signal, allowing you to do new feats of immune engineering. In the consideration for COVID vaccine development, a number of important principles apply. The first is the selection of an antigen to raise antibodies or T-cell responses, which can be based either on the whole organism, subunits of the virus, or epitopes. Also important, bottom left, the choice of adjuvants, because you want to promote activation of the innate immune system to present the antigen. And then also the vaccine needs to be in a format where it can be effectively be delivered to the antigen presenting cells. So the shape and size of that nanoparticle is important. And then lastly, from the perspective of scalability in the current pandemic, is the importance to have the technology be scalable and manufacturable. So vaccine basics, how do we simulate viral immunity? Well, if you then allow me to introduce the coronavirus nanoparticle, which via its spike protein enters epithelial cells via the ACE2 receptors into an endosomal compartment where the positive RNA strand is released, translated into proteins leading to viral assembly, and then that virus breaks out to infect a new population of cells, including top right cells such as antigen presenting cells in the immune system, where the viral proteins are presented via MHC gene products to T helper and B cells to make anti-corona antibodies, which prevents viruses from binding or tagging them for destruction. In addition, important promotion of cytotoxic T cell responses, which have the important function of destroying infected T cells and ultimately clearing the virus. And then also embedded here is the importance from a vaccine perspective of long-lived memory B and T cell responses to give durable immunity. So from the perspective of COVID vaccines, we are drawing on contemporary vaccine technology, live attenuated viruses, inactivated viral vaccines, and viral vectored vaccines, which comprise about 40% of the current approximately 170 different vaccines being cataloged by the WHO. What I'm going to focus on today is the group on the right-hand side, the next generation nano-enabled vaccines to deliver DNA, RNA, and then proteins or peptides. These comprise approximately 60% of the vaccines currently being developed. So I'm not going to say much about the conventional vaccines, but a word or two about the live attenuated and inactivated viral vaccines. Here we use the whole pathogen, weakened or killed. An example of the weakened virus is the codagenic intranasal vaccine that is currently entering clinical trials. These viruses provide highly potent humoral and cellular immunity with the advantage of being able to do so by single dose administration. There are some safety concerns, none of which have emerged as yet in the test so far, but they could revert to the pathogenic form. They could reactivate in immune deficient people, and there's also the possibility of a recombination with circulating wild-type viruses. A downside is also that these vaccines need to be distributed in refrigerated fashion. Several COVID vaccines also draw on inactivated viruses, for instance, the vaccine being developed by Sinovac. These vaccines are theoretically safer, but perhaps less immunogenic. They require multiple dose administration and often use an adjuvant. A Sinovac vaccine uses alum, which I mentioned to you earlier on. Important data published recently for these viruses, for instance, publication in Science on the Sinovac vaccine. Just as far as publications are concerned, I will tell you now that all of the work that I will mention here have been published in the last 3 months, some of which are not even out yet in its final published format. The viral vectored vaccines use live viruses in which some genes have been inactivated to provide replication. So as you can see on the right-hand side, inclusion of a coronavirus spike gene that's delivered as a viral DNA provides the antigen in this case. The choice of often non-replicating adenoviral vectors, type 5 adenoviral vectors, for instance, CanSino vaccine, the Beijing Institute and Canadian Center. Oxford AstraZeneca uses a chimpanzee adenoviral vector in their vaccine. The advantages are broad tissue tropism and inherent adjuvant properties, as well as scalability. Potential disadvantages is pre-existing immunity to adenoviruses in the population may dampen the efficacy, and that there's some of these viruses have limited packaging space for long nucleic acid sequences. Most recently, 2 publications on the CanSino vaccine and the Oxford vaccine have been published in Lancet I will indicate to you that the Oxford vaccine in the last 24 hours, the phase 3 clinical trial, has been halted because of a putatively a severe side effect in one of the participants. What I will focus on is the next generation nano-enabled vaccines. In this case, we use nanoparticles, lipid nanoparticles made of polymers, liposomes, or on the right-hand side, viral-like particles for DNA delivery, RNA delivery, or delivery of protein-based vaccines, subunits, peptides, etc. The advantage here, of course, is that we have viral mimicry, that we can entrap and protect bioactive molecules such as the nucleic acids. Antigen-presenting cells prefer the particulate antigens. Multiple cargo loading. Antigens, nucleic acids, and adjuvants can all be combined in the same particle. Control over biodegradability. Control over biodistribution and lymph node targeting. Control over the intracellular release and presentation by the major histocompatibility complex, and scalable manufacturing. Nucleic acid vaccines is a novel vaccine concept, not yet FDA approved. The advantages using nucleic acids is speed, scalability, and reduced cost. To deliver DNA, there is the additional requirement that electroporation be provided. That's a physical chemical force for uptake. Whereas for RNA, it's easier because just viruses or particles can perform that function. All antigens are delivered in a format of a natural antigen, so it's easy to present to the immune system to generate antibody responses as well as activate CD4 helper T cells and CD8 cytotoxic T cells. Another advantage is that the nucleic acids have intrinsic adjuvant activity. So example of DNA vaccines is that of Inovio Entozydos, and examples of the RNA COVID vaccines are Moderna, Acutus, Arcturus, and Pfizer vaccine. There is a theoretical advantage of mRNA over DNA in that RNA vaccine only needs to enter the cytosol. There's no need to go into the nucleus for genome integration. It is therefore not necessary to develop it as a gene therapy as is required for DNA. RNA is transiently expressed with quick degradation, fast to synthesize and purify, and could be done at a lower cost because there's no cell or protein requirements. I just want to say that the RNA can either be delivered just as a single strand as non-amplifying vaccine, or can be delivered as a self-amplifying construct where the RNA is translated for both the antigen as well as some viral replication machinery to amplify the amount of RNA that is being expressed. So quite ingenious design has then gone into developing these nano-enabled vaccines, which I will briefly describe to you. At the top left-hand side, I will describe the important design of the antigen. Quite ingenious in what has been done. In the middle, the importance to take the nucleic acid and encapsulate it into a nanoparticle, as done by Moderna, Acutus, or Pfizer as examples, which makes use of specific lipid technology, cationic lipids, as well as structural lipids, to fold the nucleic acid in there. And then on the right-hand side, after you've developed the particle and it's taken up by the immune system, it enters, like the virus, in an endosomal compartment, and you need to get the RNA out of there. So a lot of technology also gone into release that RNA to make the antigen without damage to the cell. So let's start with the antigen consideration. The important lessons come from MERS and SARS and the importance of the spike protein as an antigen for raising neutralizing antibodies. SARS, of course, is the severe acute respiratory syndrome coronavirus. I'm also going to call it COVID-1, emerged in 2003 with a case fatality rate of 14 to 15%. MERS is the Middle Eastern Respiratory Syndrome coronavirus, emerged in 2012 with a case fatality rate of 36%. In both of these, the S protein is responsible for receptor-mediated recognition and membrane fusion. The S protein and its domain was also earmarked as target for making neutralizing antibodies, and then in the subsequent era of making the COVID-2 vaccine, the smart design of that S protein to present it in a formation that is highly immunogenic has also been quite ingenious in terms of the engineering that took place. Okay, so let me explain what was done to derive a highly immunogenic S protein. On the top left-hand side, the spike protein is the target for a lot of immunotherapy. I just want to remind you that nucleocapsid membrane and envelope proteins are also important, and I will address that when we talk about T-cell responses. The coronavirus, on budding from the cell and entering the new cell, makes use of the spike protein, which undergoes a proteolytic process in order to activate it. The gene for the spike protein, when it was cloned, revealed that it consists of an S2 as well as a trimeric S1 subunit. It is the S1 subunit, shown here, that binds to the ACE receptor in green for viral uptake. When the crystallographic structure of the S protein was published, also became clear that the S1 subunit, that it is a specific domain of the S1 subunit that binds to the ACE receptor. This is also known as the receptor domain binding subunit, or RDB. The RDB in the virus can lie on the surface of the S1 subunit, but in order to enter cells, it requires that it stands up. And in the stand-up, the pre-activation by a protease, furin, allows the subunit, one subunit, to stand up and engage the ACE receptor. In the subsequent series of events, once one subunit is bound, a second stands up, and then a third. Ultimately, the virus is pulled into the cell, whereupon the S2 subunit is strung out into a peptide string. Now, why I'm telling you this is that in this conformation of binding, most of the neutralizing epitope sits underneath the hinge region of the stand-up S1 subunit. And this is sort of a viral protection mechanism where the virus wants to hide them and only at select moments open up for immunogenicity. So a lot of design has gone into mutational alteration then of the S subunit to present it in an immunogenic form. And this was done for some of the vaccines by providing mutations in the furin-sensitive cleavage site, as well as mutagenesis of two prolines to allow the S1 subunit to be engineered into a permanent stand-up fashion, as shown in this negative state EM. Here you can see the three subunits standing up in the SARS and the MERS mutational antigens compared to the wild type, where they are both in the pre and post-fusion complexes. With the RNA in place, important packaging needs to be undertaken. I want to just say that once the sequence of the RNA was refined for both the conventional and self-amplifying replicons, a lot of work has also gone into making that backbone of the RNA chemically modified to provide superior protection as well as to promote adjuvant effects. And that was done through a number of engineering interventions, such as chemically modified uridine and 3' and 5' untranslated regions. Examples of the conventional RNA vaccines is from Moderna and Pfizer, for the self-amplifying or the replicon vaccines, Acutus and Arcturus. With the RNA in place, the design of the nanoparticle was equally smart. The design feature is for a lipid-based nanoparticle. Lipid-based nanoparticles have been some of the best described materials in nanomedicine, for instance, liposomes, where the lipid components, the structural lipids, cholesterol, phosphatidylcholine, is chosen because they have a cylindrical packaging arrangement that allow them to form lipid bilayers. To make the vaccine an additional cationic ionizable lipid with a large head group, a linker, and long hydrocarbon chains was brought in to have an arrangement into a cone-shaped lipid structure, which does not want to easily enter a bilayer but folds up into a penny roll. That's also called a hexagonal phase. In order to design this nanoparticle, the RNA is infused into one flow stream and the ethanol lipid phase into another flow stream in another important technology that has been developed alongside nanotechnology, namely microfluidic mixing. This allows high throughput construction where first the cationic lipid binds the negatively charged RNA. The particle then puts out its hydrophobic tails to make an inverted micelle. Once this happened, with the increased polarity of the solution, polar lipids coat the particle surface and ultimately coalesce into an electron-dense core with a structure surrounded by a lipid monolayer that contains cationic lipid. And the big advantage is not only to bind the RNA, but where the particle now enters, like the virus, the endosomal compartment, these cationic lipids will find their way towards the endosomal membrane to make a hexagonal structure, which actually results in the nanoparticle lipid fusing with the cellular lipid membrane, releasing the RNA. And this process is also sped up by the fact that the cationic lipid turns on a proton pump inside the endosome, which imports protons and a lot of water to push out the RNA. The other big advantage in the design is that the nucleic acid robustly induce adjuvant effects at the injection site. And that is derived from the fact that both the nucleic acid in their design, as well as the cationic lipids, at the site where you inject it, as you see in the bottom right, in the muscle, generates inflammation. The nucleic acid and the cationic lipids act as danger signals to the immune system, which signals via a number of sensors. I'm not going to go through all of them, but the toll receptors is an example. These sensors are responsible for cytokine and chemokine production, which presents inflammation that recruits antigen-presenting cells. The same innate immune signals then is also responsible for the maturation of those antigen-presenting cells by stimulating, for instance, MHC gene processing, as well as antigen presentation. Altogether, the nucleic acids also have a very important signal in the direction of the immune system. That's a so-called Th1 stimulus. I'll explain that in a moment. The particles are designed of a size to easily enter the lymphatic drainage space. So the importance of everything that I've told you now in terms of nano-enabled vaccines is very important as a means of trying to promote that herd immunity. With Graham recently cautioning that if we leave herd immunity solely to natural infection, even at a 1% mortality rate, the worldwide mortality could be 40 million people. I'll explain the subunit vaccines to you in a moment as well. Okay, so what has been the experience then from MERS and SARS and using the nanoparticle vaccination, the MERS and SARS vaccine, and using the S protein for vaccination. Animal studies have shown quite effective generation of viral neutralizing antibodies to the S protein or the RDB that blocks viral binding and membrane fusion with protected immunity. However, and this is not widely known, but in an attempt to develop those vaccines earlier on, it was noticed that when ultimately after vaccination, the animals were infected with COVID-19 and MERS virus, severe immune side effects occurred at the lung. So side effects in terms of the time of infection is important, and the choice of vaccine components and adjuvant during the design phase is critical for avoiding these side effects. I'll briefly tell you that there are two major types of side effects that can occur at the time of viral challenge. One is antibody-mediated enhancement of infection, and the other is a cellular-mediated event that requires a certain type of T cells and eosinophils to induce severe lung immunopathology. The antibody side effects seem to be in vaccine components where the virus, where the immune response generate non-neutralizing antibodies. These are often of an IgG subtype, and in this case, instead of neutralizing, the antibody promotes uptake into antigen-presenting cells and more viral growth. Second immune side effect is where the FC binding to the virus introduces the virus to an endosomal compartment where it provides the danger signals that I described earlier on with cytokine and chemokine production. The cellular pathology with eosinophilia first detailed when a vaccine was developed for respiratory syncytial virus in children, where some mortalities occurred. In several MERS and COVID vaccines, there was post-vaccine enhancement with severe eosinophilic lung damage across multiple animal models from mice to monkeys. And important that it was noticed that side effects tended to decline with the use of the S protein receptor binding domain, or if you provide Th2 skewing adjuvants. Okay, so I promise you, I'll tell you about Th2 skewing adjuvants. When the immune system is switched on by antigen presenting cells promoting T cell responses, there are a number of different directions in which the immune response can develop. For vaccination to a virus, we want to have a Th1 mediated immune response, which makes use of Th1 CD4 T cells, CD8 cytotoxic T cells in an environment where Th1 cytokines such as interferon gamma promote the immune response, also responsible for B cell class switching to make IgG2A antibodies. Compare that to the Th2 immune response, where the predominant cytokine environment is the IL-4 and the IL-5 that drive in the eosinophils and promote B cell class switching to IgG1. So from the MERS and COVID-1 examples, turned out that the reason for the severe side effects was unbalanced immunity with Th2 favored over Th1 immune development. The possible explanations were the choice of viral components, that nucleocapsid protein was more prone than RDB. Also the use of Th2 adjuvants, alum, promoted Th2 skewing. Aging also played an important role. So in order to prevent that, important design is then to provide Th1 skewing. And I've already described to you that that's inherent in the nucleic acid backbone. The choice for S protein and RDB, as well as the possibility to use a number of adjuvants that specialize in providing Th1 skewing, such as inulin polysaccharides, CpG oligos, the aluminum oxyhydroxide nanorods that I mentioned to you earlier on, as well as a number of lipid-based toll receptor agonists. So what is the finding. I'm going to choose one vaccine, to the Moderna vaccine, delivering the mRNA 1273 that provides that pre-fusion complex for the S protein. Vaccination experiments in animals showed highly effective induction of binding antibodies, most of which were neutralizing antibodies in the bottom, decreased the viral load, and occurred in an immune environment where there was a balanced promotion of IgG1 versus IgG2A responses. And in the spleens of the mice, on the top right-hand side, a type of a T cell that is a T helper that is Th1 skewed by virtue of the fact that there was more interferon gamma than IL-4. Interesting, as a control in these experiments, they also used an whole S protein with alum as the adjuvant. That led to an unfavorable, unbalanced immune response in which there was a dominance of Th2 immunity. When the same vaccine was tested in human subjects, the vaccine was administered in the deltoid muscle on days 1 and 29. There was rapid seroconversion by day 15, with high titers of IgG antibodies being produced to the mutagenized antigen. These correlated with high titer antibodies to the RDB domain, viral neutralizing titer antibodies on the right-hand side, and from the peripheral blood lymphocytes, a preponderance of cells that produce interferon gamma and interleukin-2. That means they are Th1 skewed. Moreover, in terms of safety testing, there were few side effects, and where side effects occurred, they were mostly minor or of moderate severity. So what I'm going to do now is to tell you that in the effort of vaccination now in humans, what have we learned from humoral immune responses from people that were naturally infected? And these confirmed the immunodominance of RDB and viral S protein in protective immunity. Most symptomatic COVID-2 patients make neutralizing antibodies, but 33% of patients have a poor neutralizing antibody titer. Anti-RDB IgG in convalescent serum shows a promising correlation to protection. The RDB-based ELISA assay is potentially useful for identifying plasma donors. Some IgA and IgM were also detectable but short-lived. I want to point out, however, that IgA is also produced at mucosal surfaces, so we should not overlook the importance of that. The final comment that I want to make is that the antibody responses were not detectable in all human subjects, especially in less severe infections. And that brings up the question, are they unprotected, or do T cells make a difference in this environment? Before I tell you about T cells, let me also introduce vaccine durability, which received a lot of attention. Antibodies against COVID-1 and MERS seem to last for at least 2 to 3 years. In contrast, antibodies to seasonal cold viruses only provide short-term responses, allowing homologous reinfections. Early indications from a study published here from UCLA by Francesco Ibarondo and Otto Yang indicate that COVID-2 RDB serology may have a short half-life of 36 days. That needs to be confirmed by more studies, but the current expectation, and I hope this turns out to be correct, is that vaccines that generate neutralizing antibodies will provide a viable option to COVID-2 herd immunity. However, I want you to recall that while neutralizing antibodies block viral entry, CD8 T cells are required to eliminate infected cells and viral clearance. T cells confer durable immunity against conserved viral antigens, so viral antigens other than S protein also play a role. And we've also learned from durable vaccine responses in smallpox and measles which acts as benchmarks for vaccine development, the importance of B-cell memory with the assistance of T-cell help. So this conjures up important questions. How much emphasis is needed on vaccine strategies for T cells, memory B cells, and conserved viral regions? Can memory T and B cells protect in a setting where there's an acute viral disease with a short incubation period? So to address T cell immunity, in another one of these recently emerged publications from Sakin et al., addresses the importance of memory T cell development and persistence. In the Swedish cohort studies, the presence of COVID-2 specific CD4 and CD8 T cell responses during the acute infection was correlated with a high number of activated cytotoxic T cells. In the convalescent phase, also the expression of polyfunctional and durable memory T cells with a CCR7 phenotype. Importantly, the durable and functional memory T cells were also detectable in convalescent subjects with asymptomatic infections or exposed seronegative family members. So even if you have a low-grade infection with a poor antibody response, you still make T cells. Much of the memory T cell response was directed to nucleocapsid and membrane protein in addition to S protein. Similar findings was published last month by Lebert et al. So from the perspective then of T cells, I'm now going to introduce for you the protein and peptide-based vaccines and viral-like particles, because these are the technologies that we will rely on if the other vaccine types do not give durable immunity. The choices here is to use nanoparticles or viral-like nanoparticles that deliver minimal structural components of the COVID-2, such as the intact S protein, S1, S2 subunits, the RDB or peptides. Examples include the vaccines being developed by Novavax, Sanofi, Johnson & Johnson. Multiple particle types are being used: liposomes, polymers, lipid nanoparticles, and then very important also the technology of virus-like particles, which are constructed by mammalian, insect or plant viral proteins that self-assemble to make nanoparticles that deliver RDB or other subunit components. One example is the iBio tobacco plant virus particles, which are comprised of self-assembly proteins that is produced by a high-throughput technology known as fast eye farming. The viral-like particles are generally very potent. All of the above formulations are frequently formulated by using non-nucleic adjuvants. And then importantly, and I'll show you an example, can also be designed to put a ligand on to target specific areas in the lymph node, not just the lymph node in general. These potential future vaccine effort benefit a lot from the current technology for epitope mapping, as well as informatics to identify B and T cell epitopes or combination of B and T cell epitopes that can be used, and is currently being used by Oncogen and Synvax for vaccine development. Importantly, Grafoni, Ahmed, Gautier, all recently published papers that have promoted our understanding of and mapping of multiple B and T cell epitopes for the COVID-2 S1 nuclear protein and conserved viral epitopes. The work identify that a lot of these epitopes are shared by CoV-1. The B cell epitopes are potentially helpful from a perspective that you can give them to anybody. They're universal because they do not require HLA's restriction. Where you introduce HLA restriction, then of course only certain people in the population will benefit. But both Ahmed and Gautier has now shown through the technology that is available that you actually can identify T cell epitopes that can associate with multiple MHC alleles. In the case of Gautier, they predict that they can provide vaccines where for 5 or 6 vaccine peptides, you can immunize everybody The advantage of epitopes in vaccine development is that they are relatively inexpensive, scalable, safe. You can use them to generate humoral and T-cell based immune responses, or a combination of B plus T-cell immunity. And there's currently a lot of efforts, informatic-based efforts and updates that can provide iterative vaccine design. So I cannot tell you more about these vaccines at the moment. They are in development. I'll show you one example of a vaccine that we're trying to develop here in CNSI, where we've used a polymer nanoparticle, which we've packaged with viral subunits or epitopes, inclusion of a very strong Th1 adjuvant, and where we can also provide targeting sequences. When this was done in collaboration with Dr. Ibarrondo as well as Otto Yang, when we injected mice on 3 different occasions and followed that by a blood withdrawal. On day 21, the vaccine delivering the Th1 adjuvant had a very strong IgM response to the subunit, but no IgG. The IgG emerged subsequently and seemed to be stable and long-term presentation. So I cannot tell you more about this. This work is in development. What I do want to speculate on, based on what I told you about nucleic acids, and what I'm telling you now about epitopes, is the possibility that what we may see is the development of mini-gene nucleic acid platforms for multivalent vaccine constructs to generate B and T cell epitopes for scalable nanoparticle manufacturing. In short, technology known as string of beads have been developed approximately 15 years ago, which allows epitope selection, for instance, for an infectious agent, with careful flanking sequences that allow you, through making mini genes with spacers, to present a small piece of nucleic acid that, if cleaved in the correct way, can present the antigen presenting cells with multiple epitopes that does not need to be of a peptide origin. Interestingly, Yamarkovich, and also one of these recent reports, have synthesized 65 33-mer peptide sequences to diverse B and T cell epitopes that can be used in tandem with Th1 adjuvants. The epitopes were selected to be for evolutionary conserved viral elements, plus more variable regions such as the S protein, to present diverse HLA presentation, to stimulate CD4 and CD8 T cells, also to include linear and conformational B and T cell epitopes, and also selected with a maximum degree of dissimilarity between the viral immunopeptidome and the human sequence for maximum immune responses. So string of bead vaccines have been shown experimentally to be successful for CMV and influenza, and I will not be surprised if we see this technology development in future, should the other vaccine types not provide a durable immunity. So to end this part of the talk, I'm going to just say briefly that one of the critical vaccinology needs that have been defined in vaccine development is to get to a specific place in the lymph node where B cells receive instruction from a memory T cell, a CCR7, positive memory T cell type. That is the CEIN study. So these are the types of T cells that develop. Because these types of memory T cells, they're also known as follicular helper T cells, or TFH for short, when entering the germinal centrum, provide class switching and affinity maturation to enable B cell memory to develop. And these memory B cells emigrate from the follicle to provide stable and a long-term memory B cell pool. in what I find a fascinating development, Pardee et al., in developing the lipid nanoparticles that deliver messenger RNA antigens for Zika, influenza, and HIV vaccines, have shown that these have a potent effect on promoting the THF and germinal center cooperation with durable neutralizing antibodies. And these results are also corroborated in rhesus monkeys by an influenza HA vaccine that was delivered by a lipid nanoparticle. How exactly the lipid nanoparticle does that is not known, but I suspect that it is due to the fact that these antigens are all glycoproteins. And so from that perspective, the laboratory of Darrell Irvine, working on developing a vaccine for HIV GP120, which is also similar to the S protein, a fusion protein, has shown how important glycosylation is. If you use the HIV GP120 to vaccinate an animal, as you see in the confocal microscope example here, in the lymph node, most of the GP120 only enters the subcapsular sinus. This is the smudge of red stuff that you see here. If, however, the GP120 is incorporated in a nanoparticle that has a polymerizing protein that is rich in glycans, there is a high degree of entry of the antigen into germinal centers where there's follicular dendritic cells driving germinal center B cell development. And the trick here, according to Darrell, is that the glycans trigger mannose binding lectin that activates the complement cascade. And it is the complement cascade that is then involved in generating the immune response. He's also shown that the glycosylation features applied to HIV GP160, influenza hemagglutinin. And then importantly, many of the current messenger RNA encoded proteins that have been chosen for antigens that are glycosylated. So what I predict is that the homology of the CoV-S protein with related class 1 fusion proteins in influenza and HIV also holds great promise for future vaccine design by considering the pre-fusion conformation that I've already described, as well as the glycosylation status. It's interesting that the COVID-2 influenza and HIV envelope contains many oligomannose glycans that are important in antibody generation. And I will just remind you again that IgG2A is generated more frequently against carbohydrate antigens than protein antigens. My last slide on vaccines is then also to just say that an interesting development is that vaccine-induced natural selection may impact maintenance of immunity in case of pandemic persistence. At the moment, there's very little evidence of reinfections for COVID-2, in part because the COVID-2 genetic proofreading is excellent and provide for low sequence diversity. However, natural selection can, on rare favorable occasions, result in mutations such as documented for COVID-1, where an RBD mutation led to escape from neutralizing antibody efficacy. Importantly, in a recent paper published in Cell by Dr. Korber et al., there was depiction that for COVID-2 and S domain mutation in glutamate 614 to aspartate occurs in the S1 region where it interacts with S2. This is the furin cleavage site. And where this mutation developed within a time frame, is the top right-hand side, of 1 month as the virus emerged from Wuhan to Europe. Patients with the mutation shed more viral RNA compared to the wild-type virus with significant higher infectious antibody titers. So pandemic persistence could potentially lead to the accumulation of immunologically relevant mutations in the populations as vaccines are introduced. And I encourage you to go and look at the website that Dr. Korber has designed, where they developed informatic tools to act as an early warning strategy to evaluate spike protein evolution during the pandemic. My last two slides will then say that nothing is isolated in the world of nanomedicine. All these engineered designs that I've now shown you that are used for COVID can also be employed and in fact originated from raising vaccine or antibody response, antibody and cellular immune responses against tumor antigens. Tumor antigens is shed, educated in the regional lymph node and return to the cancer site, where intervention with lipid-based nanoparticles have been able to show to drive important responses, including immune responses. Our own contribution to this is to develop an endogenous vaccination response, which is premised on the fact that if we introduce nanoparticles that deliver certain types of chemotherapeutic agents, top right-hand side, induce a form of apoptosis on the bottom right-hand side, where the dying tumor cell expresses an eat-me signal in the form of calreticulin, plus two adjuvants, HMGB1 and HTP. And that occurs on the background where the major breakthrough in checkpoint blocking antibodies have identified that these antibodies could be highly effective in subpopulations of certain cancer types. And the people that do seem to respond to checkpoint blocking antibodies are those that have an immune system already loaded with cytotoxic T cells, so-called hot. And the 80% who do not respond, expressing a cold immune system where the CTLs are not present. So we hypothesized that some of these chemotherapeutic agents then can be used to provide endogenous vaccination to turn the immune system from hot to cold. And in order to do that, we've developed a series of technologies. I'll just show you one example on the left-hand side, a nanoparticle that have a hollow porous interior with a lot of packaging space of drug covered by a lipid bilayer to look like a liposome, as you see on the right-hand side, but much more effective in the amount of drug that can be loaded and its ability to deliver that to the cancer site. If we take the case of a pancreatic cancer, an orthotopic pancreatic cancer that simulates the process in humans, you can see that treatment with saline or free irinotecan, one of these chemo agents that drive a vaccination response, had very little effect. An irinotecan liposome was effective in decreasing metastases and the primary. The silicosome, very effective elimination of the metastases and a much shrunken primary tumor. Moreover, also demonstrated that in these dying tumor cells, there is activation of calreticulin and the immunogenic cell death pathway, infiltration with cytotoxic T cells, indeed turning the tumor hot, and then amazingly also expression of anti-PD-L1, which then allowed us in a endogenous tumor vaccination response to demonstrate that combination of the irinotecan delivering nanoparticle with anti-PD-1 antibodies lead to a strong immune outcome. So of course, all of this work requires the expertise and cooperation of many, two associate professors, Meng Wan and Chan Jia, postdoctoral fellows, Jiansheng Liu, Casey May, Sean Allen, QC Liu, staff scientists and students all contributing to this, and then also the work done in CNSI, where our multi-team efforts in nanomedicine and other areas of nanotechnology against the mantra of basic discovery leading to translation and commercialization has allowed CNSI to establish a very successful entrepreneurial platform where 35 companies are now incubating in our space, and also the development of a very successful proposal facilitation unit where the number of applications written is $240 million for the last 3 years, with a 26% funding rate. That is much higher than what you currently get. Thank you very much.
Thank you so much, Andre. That was outstanding, quite a tour. So if anybody has questions, please put them in the Q&A function, and I will read them to Dr. No. Andre, I have a question. Is there a difference in the T cell versus antibody response or immunogenicity between more conventional vaccines and the nano-enabled vaccines?
Jody, conventional vaccines is a whole spectrum of vaccines that goes from anywhere from using infectious agents all the way down to subcomponents of infections. And there is, depending on the type of infection, good evidence for the contribution of antibodies as well as T cells, but with various emphasis on the contribution of each of those to the outcome. For simple subunit vaccines that is generated with, say, a bacterial or a viral component, there's very good evidence that antibody responses there plays an important role to protect. But as you get to more complicated infections, measles, smallpox, there is a progressive reliance also on the involvement of T cells and memory B cells in the sustaining of that response. And I think in terms of what you see in the current era with all the information going around, it is actually quite complex. And I think that our aim at the moment is we're going for viral neutralizing antibodies as the primary reliance. I hope it is successful, but I suspect that what we're going to find is that for durability of the immune response, T cell responses are going to be important. Let's hope that the current batch of vaccines also drive durable T cell responses to make that long term. If not, we will then have to take apart all the potential contributing components into the type of design that I've shown you here, where you have to dynamically mix and match B and T cells, as well as promotion of specific types of immune responses at germinal centers in order to carry long-term immunity.
Got it. Thank you. We do have a few questions here in the chat. One is, what sorts of adverse reactions are being seen to the COVID vaccines?
Well, so far, remember, we're in phase 1, 2, and in a few places in 3. Most of the adverse reactions at this stage seem to be reactions that are of an inflammatory nature. That means anywhere from pain to the infection site to fever, chills, headaches, which is quite common with vaccinations similar as flu. However, we have not entered into the era yet where after vaccination people get infected. That was where in the case of the MERS and COVID-1, it was noticed then that at the time of infection, people develop these severe ADE and cell-mediated immune reactions. At the moment, very good sign that there seems to be good evidence in the few cases presented so far of a balanced T cell response with Th1 skewing, therefore predicting that we will not have ADE or cell-mediated immunity, but the proof lies in the pudding. That's why the current importance of the safety of vaccines is such an important issue before we move ahead.
Great. All right, I think we have time for a couple more, or at least one more. This from Jeffrey Miller. To target lymphoid germinal centers, does the antigen need to be specifically glycosylated, or can a nanoparticle containing antigen be targeted by linking appropriate glycans to its surface? And he also said, great talk.
Well, I think that I tried to show you by virtue of one example from Dr. Darrell Irvine that if you choose the antigen to be glycosylated and you package it in a nanoparticle, you will accomplish the ability to, if you make the particle design of the correct size, of the correct polarity, etc., that you can get it to the germinal center. I think a lot more efforts will probably now be directed towards that type of engineering. But my suspicion, and this is just a notion, is that yes, that glycosylated antigens, I think, is going to perhaps will provide one of the avenues of successful generation of memory B and T cell responses for the reason that I explained.
Thank you very much, Dr. Nell. There are a few more questions in the question and answer. I encourage people to go ahead and email Dr. Nell specifically because we are running out of time. Thank you all for your attention. It was an outstanding grand rounds. Thank you very much, Andre.
Thank you, Jody. Thank you, everyone. Keep safe.